Good afternoon, everybody, and welcome to Pediatric Grand Rounds. My name is Sandrine Van Schaik. I'm the Vice Chair for Education in our department. And I want to say happy Yom Kippur, but I hope that everybody's actually celebrating Yom Kippur is not currently listening, but maybe you will watch this on video later. It is my great pleasure to introduce our speaker for today. Here you have actually the upcoming presentation for next week, but for today, we are going to listen to Dr. Beernink talk to us about meningococcal B vaccines, talk entitled From Genomes to Gonococci, which, when I first came to this country, it took me a long time to know how to pronounce that. So I got there, I think. Dr. Beernink is Professor of Pediatrics in our Division of Infectious Diseases and Global Health. He holds the Clorks Endowed Chair of Immunobiology and Vaccine Development, and his research lies at the fascinating intersection of vaccinology and structural biology. Using sophisticated techniques like X-ray crystallography, his lab designs novel vaccines against challenging bacterial pathogens. In 2021, he received the prestigious Scientific Career Achievement Award from UCSF Benioff Children's Hospital, Oakland, where he has been working for 2 decades. His presentation, as I announced by the title, will overview the meningococcal B vaccine, tracing its cutting-edge development and its surprising potential to also protect against gonorrhea. So please join me in welcoming Dr. Peter Beernink.
For the introduction. I suspect that introduction was written by AI, so don't be surprised if it's not completely on target. Good afternoon, and thanks for the invitation to present today. I'm going to present on the topic of meningococcal serogroup B vaccines, how they were developed using genome sequence information, and how they led to progress towards a gonorrhea vaccine. First, here are my disclosures. So first, I want to summarize the epidemiology of meningococcal disease in the last 25 years. The epidemiology varies geographically, and in the US, there's an historically low incidence of just below 0.1 cases per 100,000, and cases have been declining since the early 2000s. See if this pointer works. To remind you, the serogroups are defined by the capsular polysaccharide. Group B has been responsible for the most cases in the past 20 years, and there's been a recent uptick in serogroup Y cases, seen here. Risk factors include complement deficiencies and social factors such as communal living, going to bars, kissing, and smoking, and these social factors are frequently co-occurring. There are relatively high asymptomatic carriage rates, about 10 to 20%, and therefore high levels of natural immunity. There are 2 peaks of incidence by age. The first peak in infants and toddlers seen in the leftmost bar here, and a second smaller peak in older teens and young adults seen in the middle of the graph. Graph on the right shows older data only for serogroup C, where the incidence is shown in blue, and the incidence is inversely related to the presence of naturally acquired antibody, with serum bactericidal activity shown in green. And the serum bactericidal activity decreases after maternal antibody wanes and increases with increasing age. And most adults, or most people above the age of 10, have naturally acquired antibody and immunity. Let me briefly explain the serum bactericidal antibody assay, which I'll be referring to throughout my talk. This is an in vitro assay to measure killing of the bacteria. We mix antibody, typically serial dilutions of test serum, bacteria at a known concentration, and complement, usually human serum frozen shortly after clotting. We incubate the mixture, plate on agar, and count colonies the next day. The bactericidal titer is the serum dilution that gives 50% survival. I'm sorry, lost my cursor. Bactericidal titer is the serum dilution that gives 50% killing compared with a no antibody control. And a serum bactericidal antibody titer greater than or equal to 1 to 4 correlates with protection from early studies in the 1960s. And this is an example of a killing curve. So 50% survival gives a titer of about 70 in this example. There are two classes of meningococcal vaccines. The first is protein polysaccharide conjugate vaccines, which include the quadrivalent Men-ACWY vaccine introduced in 2005. These vaccines contain polysaccharide antigens from the respective serogroups coupled to a carrier protein, such as diphtheria or tetanus toxoid or CRIM, which is a mutant diphtheria toxin. These vaccines have a category A recommendation from ACIP for 11 to 18-year-olds. Sorry, I lost my cursor. Second category is protein-based vaccines against Men-B, and these were licensed starting around 2013. These vaccines contain protein antigens and or outer membrane vesicles, or OMV. These vaccines have been issued a category B recommendation or shared clinical decision-making for persons aged 16 to 23 and at higher risk, for example, people with complement deficiencies or in outbreak settings. The newest meningococcal vaccines are pentavalent vaccines that combine the two vaccines I just talked about, a conjugate component against ACW and Y, and protein components against Men-B. These were licensed starting in 2023 and have a complex recommendation. Men-Penta vaccines may be used when both Men-ACWY and Men-B are indicated at the same visit for the following groups. 1, healthy persons aged 16 to 23 years, the routine schedule, when shared clinical decision-making favors administration of Men-B vaccine, and 2, persons aged greater than or equal to 10 years who are at increased risk for meningococcal disease, for example, because of persistent complement deficiencies, complement inhibitor use, or functional or anatomic asplenia. So you can see this is kind of a nightmare and why some people don't like shared clinical decision-making. I want to talk about the development of vaccines against Men-B. Here, the conjugate vaccine approach was not feasible because the group B polysaccharide, polysialic acid, resembles host glycans, is therefore poorly immunogenic, and carries the risk of eliciting autoantibodies. One experimental approach has been to use chemical modification. I'm sorry, this keeps skipping ahead. Chemical modification of polysialic acid to elicit antibodies that kill the bacteria but do not bind host tissues. Outer membrane vesicle, or OMV vaccines, have been developed to combat epidemics in Norway, Cuba, and New Zealand. These vaccines are strain-specific, and they're only effective against the epidemic strain and closely related strains. There are also protein antigens, which I'll discuss more in the next slide. A search for protein antigens was done using the Men-B genome, which was published in 2000. This approach was coined reverse vaccinology because it started from the genome rather than the proteins identified in the bacteria, and has the advantage of identifying proteins irrespective of their abundance in the bacteria. This new approach was first applied to Men-B, whose genome sequence contains about 2,150 open reading frames, in other words, predicted proteins. A group at Novartis Vaccines used bioinformatics to predict surface-exposed proteins, and an army of postdocs purified about 350 antigens and vaccinated mice to identify candidates. And the criteria were that the serum antibodies must bind to the bacterial surface and elicit complement-dependent bactericidal activity. The outcome of this discovery effort include 3 protein antigens that are used in the Men-B 4 component, or Men-B4C vaccine, shown at the right. These are factor H binding protein, FHBP, Neisseryl heparin binding antigen, NHBA, and Neisseria adhesin A, or NAD-A. 2 additional antigens were fused to FHBP and NHBA, and it's not clear if these 2 antigens contribute to protection. Finally, outer membrane vesicles were added to extend protection against the New Zealand epidemic strain. And the outer membrane vesicles are depicted here. This slide shows the same 4 antigens in the MenB4C vaccine. This contrasts with the second licensed MenB vaccine referred to as MenB FHBP, trade name Trumemba, which only contains FHBP. The third vaccine, no longer available, contains only OMB, and I'm going to come back to this one later. Factor H binding protein is a key antigen in both licensed MenB vaccines. It was discovered from the genome sequence as a hypothetical lipoprotein, first described as genome-derived Neisseria antigen, or GNA 1870, by a group at Novartis in 2003. It was identified independently by traditional vaccinology by a second group at Wyeth, now Pfizer. FHBP is a surface-exposed protein that binds complement factor H, which downregulates complement activation on the bacterial surface. And antibodies to FHB serve dual functions in protection. First, they elicit bactericidal activity through the classical complement pathway. And second, block binding of factor H to the bacterial surface, which defeats the bacterial immune evasion mechanism. Despite these positive attributes, there are several limitations of FHBP as a vaccine antigen. The first is low abundance on the surface of some strains, which is shown in the graph by differences in binding of a cross-reactive monoclonal antibody by flow cytometry. Most of the strains with higher FHBP expression on the left side of the graph are susceptible to bactericidal antibodies, shown by open circles, while most of the strains with low expression are resistant to killing, showed in the filled triangles on the far right side of the graph. The second limitation is sequence variation among strains, which decreases the cross-reactive antibody. And here you can see that 9 out of 45 strains were resistant to killing by human vaccinee serum, which includes the combined effects of both antigen sequence variation and antigen expression differences. The third limitation is that binding of host factor H could mask important epitopes, or antibody binding sites. The limitation of FHBP sequence diversity is profound. The sequence variation is represented in a phylogram, which can be divided into 2 subfamilies, A on the right and B on the left. Another scheme uses 3 variant groups, where subfamily B is equivalent to variant group 1, and subfamily A is further divided into variant group 2 and variant group 3. There are about 1,500 sequence variants of factor H binding protein known to date in a protein of only 255 amino acids in length. The 2 vaccines use divergent sequences. MenB4C uses a subfamily B antigen that we call ID1, and the MenB FHBP vaccine uses a divergent antigen within subfamily B that we call ID55. The MenB FHBP vaccine also includes a second FHBP variant from variant group 3 in subfamily A, and we call this one ID45. So the questions are, how many FHBP variants are needed for cross-protection against diverse meningococcal B strains? And second, which variants are most cross-protective? We sought to address the question of cross-protection experimentally, and we selected 7 FHBP variants which are prevalent in different geographic regions, including the US, the UK, France, and sub-Saharan Africa. The MenB4C vaccine variant, ID1, is prevalent in the US, but the MenB FHBP variant, ID55, is extremely rare. and it raises the question of why that antigen was chosen. We purified the 7 recombinant proteins and immunized groups of mice. We tested mouse serum IgG antibody binding to the 7 recombinant immunogens, and we tested bactericidal activity against isogenic strains containing 7 different variants. And this use of isogenic strains isolated FHBP sequence as the only variable. I should note here that the sequence difference between the 2 vaccine variants in subfamily B, ID1 and ID55, is about 87%. So as an example, this graph shows binding of serum IgG antibody from the 7 groups of mice to the purified FHBP ID55 protein, which we put on the ELISA plate. Here, each symbol represents a serum pool from 4 mice. Anti-ID55 anti-serum has the highest IgG titers against the ID55 antigen on the plate. And the reactivity decreases gradually from right to left. We didn't expect to see this result because we thought the ID numbers were assigned arbitrarily. So when we plot the same data with sequence identity on the x-axis, we see a nearly perfect correlation between the sequence identity and the IgG geometric mean titers with a p-value of 0.003. So the explanation for why the ID numbers were not randomly distributed is that when the ID system was created, the researchers that did this took the first 15 ID numbers in phylogenetic order, and then after about 15 or so, then they were assigned arbitrarily as they were discovered. So this slide shows cross-protection measured in a bactericidal assay, as I described before. The data show a similar pattern where the ID55 antiserum gives a high titer of more than 1,000 against the matched strain with ID55, and a titer less than 10 against the ID1 strain, which is the most phylogenetically distant. The take-home message, oh, one second. And you see a kind of reverse pattern for the ID1 antiserum tested against the same 7 strains. So the bactericidal titer against the ID1 strain is more than 1,000, and this drops off quickly as you go farther in sequence. The take-home message is that in these experiments, neither the ID1 or 55 vaccine covers the most divergent sequence in subfamily B. And our hypothesis was that sequences near the middle of the phylogram, such as ID9 or ID13, should have the highest cross protection. When we compared the phylogenetic distance of each variant with every other variant within subfamily B, at the time this was about 325 variants, we found that ID9 and ID13 had the smallest average distance from every other sequence. When we tested the bactericidal activity of the ID13 antiserum against the isogenic strains, we saw the highest bactericidal activity against the closely matched strains, as you would expect, ID9 and ID13 expressing strains, and dropping off in both directions as the sequence diverged away from the center. Now I want to get back to the third limitation of FHBP, which is that binding of a host protein could mask important FHBP epitopes. A review article by Seppo Meri and colleagues stated that, quote, the use of functionally active complement inhibiting proteins as vaccines carries the potential risk that by inhibiting complement or by binding to a host molecule, they could be less immunogenic or partially masked by the host protein. To explain this schematically, when factor H isn't bound to FHBP, you generate antibodies against these neutralizing epitopes, which can block binding of factor H. And when human factor H is bound to FHBP, this creates a complex in which the neutralizing epitopes are blocked or masked. So the antibody responses are not directed towards the part of the protein that you most want to create antibodies against. Our colleagues at UMass Medical School created transgenic mice with human factor H, which allowed us to test the effect of factor H on FHBP immunogenicity. Here it's important to note that mouse factor H doesn't bind to FHBP. In two independent experiments, the bactericidal titers of immunized transgenic mice labeled TG were about 5-fold lower than wild-type mice immunized with the same antigen. What we didn't expect was that the variance among the titers was higher in the transgenic mice than in the wild-type mice. And this is not totally obvious, but you can do a statistical test that shows you the variance is actually greater. When we looked at the correlation between bactericidal titer and the amount of serum human factor H in each mouse, we saw that there was a significant inverse correlation between these. And you can see that here, the R value is negative 0.65 and the P value is significant. So there really is an effect of human factor H binding to the FHBP vaccine antigen seen in this correlation. Oh, and this is just to remind you that the mean human serum factor H concentration is about 300. And so we're in sort of a physiologically relevant range compared to humans, although the mice have a wider range of factor H concentrations than humans, which work to our advantage in this one case. In a landmark study, our colleagues at Oxford solved the crystal structure of a fragment of human factor H, shown at the top, bound to FHBP, which has implications for vaccine design. I separated the two proteins to highlight the respective interaction surfaces in blue and green. The factor H binding site of FHBP, shown in green, is oriented away from the bacterial outer membrane, which allows factor H to bind. This structure allowed us to visualize the hypothesis that binding of human factor H redirects the antibody repertoire to FHBP away from the factor H binding site, the green epitopes, and the resulting antibodies do not inhibit binding of factor H and are less effective in eliciting bactericidal activity. To address this hypothesis, we can use anti-FHBP monoclonal antibodies, or mAbs, to probe the antibody repertoire. We can compare mouse and human mAbs in their ability to inhibit factor H binding. We can elucidate structures of antibody complexes with FHBP to identify the epitopes or antibody binding sites in molecular detail. And we can complement these studies with inhibition of factor H binding by serum polyclonal antibodies from different species. This factor H inhibition ELISA shows that increasing concentrations of anti-FHBP mAbs, JAR5 and JAR3, inhibit binding of factor H to factor H binding protein in an ELISA, while mAb JAR4 does not inhibit binding of factor H. Using X-ray crystallography, we solved the structure of the JAR5 Fab fragment bound to FHBP, which showed that the epitope recognized by JAR5 overlaps with the factor H binding site, shown in yellow. Another mouse Fab that inhibited factor H binding from work by our colleagues, not shown here, bound to this top surface of factor H binding protein and totally blocked the factor H binding site. Further, the structure of mAb JAR4, which does not inhibit factor H binding to FHBP, binds to an epitope that's on the opposite face compared to the factor H binding site. So this kind of provided a mechanistic explanation for either antibodies that inhibit or antibodies that do not inhibit binding of factor H to FHBP on the bacterial surface. In a subsequent study, we cloned human Fab fragments from 3 human subjects vaccinated with MenB4C, which has the same FHBP variant used to generate the mouse mAbs I just showed you. This flow cytometry assay shows 10 human Fabs in orange. 5 of the Fabs in the middle had no effect on factor H binding. One Fab, called 5B, showed slight inhibition, And 4 Fabs in the top right quadrant showed enhancement of factor H binding. 2 control mouse Fabs were shown in aqua as controls for an inhibiting Fab and an enhancing Fab. So similarly, we solved the structures of 2 human Fabs in complexes with FHBP. The first was a human Fab called 9B that had no effect. This one had no effect on factor H binding. And you can see the epitope recognized by the antibody does not overlap with the factor H binding site. So this makes sense with our kind of model. Second human Fab called 8A was one of the enhancing antibodies, this one. And this antibody bound to an epitope that was kind of adjacent to the factor H binding site. And what we think is happening is that this Fab stabilizes a flexible loop in the factor H binding protein antigen that kind of stabilizes the factor H binding site indirectly. The review article I mentioned before further noted that, quote, the vaccine proteins would need to be slightly modified so they lose their functional activity but maintain the immunogenic potential, yet raise antibodies that neutralize the functional activity of the proteins on the bacterial surface. This is kind of confusing, so translated into English, what they're saying is that one can imagine a mutant FHBP antigen shown at the bottom that does not bind to human factor H and elicits the neutralizing antibodies that we need to inhibit binding of factor H to factor H binding protein. So we spent about 5 years on developing mutant factor H binding proteins. So this is an ELISA that shows binding of factor H to the ID1 wild type antigen shown in blue that shows good binding. And then binding of factor H to 3 mutant versions of factor H binding protein ID1. The mutants are called R41S, H48L, and S223R, which have about 100 to 250 fold lower factor H binding than the wild type. And we identified this R41S mutant by structure-based engineering approaches, and we found these other 2 mutants by library approaches and sort of modeled those into the structure from the library mutant library results. When we tested these antigens in transgenic mice that made human factor H, the 3 mutant FHBP antigens gave 13 to 20-fold higher bactericidal titers than the wild type mice. And interestingly, in contrast to the wild type FHBP antigen on the left, which I showed you before, which had an inverse correlation, the R41S mutant had no correlation between bactericidal titers and serum human factor H. And strikingly, even this mouse that had about 600 micrograms per mil of human factor H had a high bactericidal titer. So there's really no effects of factor H binding on the mutant antigen based on these data. A previous paper identified factor H binding as specific to human and some primate factor H. To investigate this further, we tested individual macaques from the UC Davis Primate Center and found that about 1/4 of the animals had factor H that bound FHBP similarly to human. And the other 3/4 of the animals had factor H with low binding. And by sequencing factor H exons, we found these differences were attributable to 2 polymorphisms in macaque factor H, which gave either low or high binding to FHBP. And for a vaccine study, we screened infant rhesus macaques to identify those with high factor H binding and matched infant macaques by gender and by age and enrolled them in the vaccine study, where we compared the wild type FHBP antigen shown in filled circles to the mutant antigen shown in open squares. This is actually, doesn't say here, This is a double mutant antigen combining the R41S and H248L substitutions that I showed you before. So you can see against a strain that has ID1, so a matched antigen, there's about 5-fold higher bactericidal titers to the mutant antigen than to the wild type antigen. Similarly, for an isogenic strain that has ID13, one of the central FHBP variants in the phylogram, with about 6-fold higher. And then as you diverge away in sequence, a FHBP ID1 strain that had about 15-fold higher bactericidal titers to the mutant antigen compared to the wild type antigen. So we really think that this is a significant advantage of using mutant factor H binding proteins. To control for differences in IgG antibody titers between the groups, we selected the 6 animals from the wild type vaccine group with the highest IgG titers and the 6 animals from the mutant vaccine group with the lowest titers, so that the geometric mean antibody titers were about the same. So really skewing this away from the mutant and in favor of the wild type by taking this subset. Comparison of the bactericidal titers showed about 6-fold higher titers for the mutant antigen, despite having removed the best animals from the analysis. The antibodies to the mutants better inhibited binding of factor VIII to the bacteria, seen by a lower ratio of post to pre-immune serum. So a lower ratio shows inhibition by the antibodies, and a higher ratio shows enhancement, as I talked about before. Finally, we looked at C4b deposition, and we showed that there was a strong correlation between the bactericidal titer on the y-axis and the amount of C4b deposited on the bacterial surface, which is a marker of classical complement pathway activity. And if you notice, almost all of the mutant data points are in the top right, and almost all of the wild type data points are in the bottom left. So there's a quality aspect to the mutant, the antibodies elicited by the mutant. They're higher quality, more factor H inhibition, and more C4b deposition. So this slide summarizes the clinical development of improved FHBP vaccines based on non-FH binding FHBP mutants and or phylogenetically central FHBP variants, addressing 2 of the limitations I talked about earlier. Each vaccine used a different platform. The first was a chimp adenovirus vector that encoded a mutant of the subfamily B antigen in the MEN-B4C vaccine. The vaccine was tested in a phase 1/2A trial, and the results are presented in this Science Translational Medicine article published earlier this year. The second vaccine is a recombinant protein vaccine that included 2 antigens, one from each subfamily, and again, not including an FHBP variant for reasons that I can't explain very quickly. This vaccine is finishing phase 2, and phase 3 studies are planned for next year. And this is proprietary data from a pharma company, so it's not published, and I don't actually have access to the data myself. The third vaccine is an outer membrane vesicle vaccine. It uses 2 FHBP variants, one from each subfamily. In this case, they did use a central FHBP variant, and the status of this one is that phase 1 studies are planned, and there's a reference to the preclinical study in this vaccines article that's in press. So I want to show you a little bit of the results from the chimp adenovirus vaccine study done by Oxford Vaccine Group. And the best sorry, this is super complicated. I'll try to just sort of cherry pick the most important data. The best comparison is the 2-dose adenovirus group, which is shown in teal, this one, and the 2-dose MEN-B4C vaccine, which is shown in yellow, both given at 6-month intervals. So after 1 dose at day 28, the titers to the MEN-B4C vaccine are slightly higher than those to the adenovirus vaccine. Comparing the teal, this is the mean, the geomean, that open square, compared to this one. So the MEN-B4C after one dose is slightly higher. And this is measured with a matched ID1 strain. However, the MenB4C, as I told you before, contains additional antigens that are present in this strain, so it's not, in fact, a fair comparison. The responses after 2 doses at day 208 are boosted in both groups, and there's the gap between the adenovirus shown in teal and the licensed vaccine shown in yellow are kind of both boosted to similar levels. And then at day 65, the antibody persists in both groups, and there seems to be a slight reduction in the difference between the two groups after a year after the first dose. So 2 points. One is this was part of this experiment, which turned out to be way more complicated than I think it needed to be, was they were testing single doses of the chimp adenovirus vaccine with the hope that 1 dose would be comparable to 2 doses of the licensed vaccine. I don't think that turned out to be the case. And then they were testing different schedules, including the second licensed vaccine. So there are a lot of comparisons and a lot of differences that it's just not the kind of controlled study I would like to see. So shifting gears, after introduction of MenB vaccines, the Neisseria vaccine field turns its focus towards developing a vaccine against gonorrhea. Gonorrhea is the second most prevalent STI and has increasing multi-drug resistance, as I'm sure you've heard. Gonorrhea is prevalent in older teens, 15 to 19-year-olds. And the rationale for using previous experience with MenB is that gonococci are closely related with 80 to 90% genome sequence identity shown schematically in the genome maps of MenB strain MC58 and gonococcal strain FA1090. And the blue box just shows a handful of antigens that have high sequence identity, greater than 90% between meningococci and gonococci. And one antigen that we worked on recently is called NSPA, and that's highly conserved between meningococci and gonococci. And there was a seismic shift in the field when Patousis Harris and colleagues published a paper on an OMV vaccine that had been used to control an epidemic in New Zealand, a MenB epidemic. The retrospective study found a 31% decrease in gonorrhea cases in the post-vaccination period, 2004 to 2006, compared with the pre-vaccination period, pre-2004. And you can see that cases were going up before introduction of the MenB this is gonococcal cases, going up before introduction of the vaccine until about 2006, and then they drop, but it's not clear. This is a retrospective study. It's not clear if this is vaccine related. So this is still an open question. So around the same time, we were developing an OMV vaccine against MenB, and our vaccine differed from the MenZB vaccine in several ways. First, our vaccine used a different vaccine strain. Second, our strain was engineered to genetically detoxify endotoxin, or LOS, lipooligosaccharide, whereas the MenZB vaccine used detergents to extract LOS. And this difference is important because detergents extract lipoproteins from the outer membrane, so the genetic detoxification approach preserves potentially protective lipoproteins, including some of the conserved antigens I showed you on a previous slide. So we performed a mouse immunogenicity experiment with our OMV vaccines and tested the anti-sera for their ability to kill gonococci in the bactericidal assay. So most of the sera gave less than 50% survival in the presence of human complement. So 12 out of 16 from the native OMV that has FHBP knocked out. This was a control for MenB, a negative control, and 11 out of 15 mice from the NOMV FHBP vaccine group had lower than 50% survival, which is kind of our threshold for significant in vitro killing. So we wouldn't have expected to see a difference between these two groups because gonococci don't have factor H binding protein on their surface. So antibodies wouldn't bind to this protein. It's an intracellular protein in gonococci. And then this aluminum hydroxide is the negative adjuvant only control. None of those mice gave lower than 50% survival in the assay. Since meningococcal OMV can elicit antibodies against gonococci, you'd expect that a gonococcal OMV would be at least effective as a meningococcal OMV, if not more so. However, gonococci have mechanisms to subvert the human immune system, so a successful gonococcal OMV vaccine would have to overcome this problem. Our colleagues working on the Men-B OMV approach I showed you in the clinical studies tested meningococcal and gonococcal OMVs separately and in combination. And here you can see that these are bactericidal titers measured against different gonococcal strains, and the Neisseria gonorrhea NOMV group and the combination NOMV group had high bactericidal titers against every strain. So this looks like it's a promising approach. I think other people have thought of it, but so far the gonococcal OMV approach has not been sort of, it hasn't gained traction. And this is the publication that's just coming out in press. So we'll see if this one goes anywhere. I think the idea here is that this combination vaccine would be the most viable to be able to prevent both Men-B and gonococcal infections. So a couple of recent papers showed that human anti-LOS antibodies contribute to killing of gonococcus. And these investigators removed antibodies to two antigens, NHBA, which I talked about before, and lipo-oligosaccharide from human vaccinee serum by absorption, and to remove, they were able to remove some of the bactericidal antibody activity. The data are a little bit messy, so I'm not showing the raw data. As I mentioned, most OMV vaccines, including Men-ZB and Men-B4C, are treated with detergents to remove LOS, but the vaccines actually contain residual LOS at about 5 to 8% of natural levels. And LOS was previously identified as a target of bactericidal antibodies going back a couple decades. So an anti-LOS monoclonal antibody called 2C7 suggests possible vaccine approaches against gonorrhea. The mAb recognizes a conserved LOS epitope, which was previously thought to include lactose residues extending from heptoses 1 and 2 in the red box. So this was the epitope based on binding of the antibody to strains with different LOS structures. This mAb elicits bactericidal activity in vitro, shown in the lower left graph. And it decreases time to clearance in the mouse vaginal colonization model, shown on the right. And this dashed line is the mAb, and this solid line is a control, negative control without mAb. And this antibody has been converted into a chimeric variant to be used as a therapeutic by our collaborators at UMass. So in collaboration with Sanjay Ram's group at UMass, we solved the structure of gonococcal OS bound to the Fab fragment of MAB2C7. And so OS, this is the oligosaccharide from LOS without the lipid A portion, and it's bound to a part of the Fab that's mostly through the heavy chain, which is the darker chain in both of these pictures. And you can see that LOS really sticks down into a cavity of the antibody. So looking at the contacts, the antibody interacts with the beta and gamma chains of OS, which are oriented pointing downward. So this is a little complicated, but the gamma chain contains this nitrogen atom that's pointing down, and the beta chain includes this lactose residue that's also pointing down into the cavity of this antibody. So the structure defines the oligosaccharide epitope, which is shown here in the lower rectangle, where this is the beta chain, that's the one pointing down. This is the gamma chain, the one kind of behind it in 3 dimensions. And this alpha chain is sort of part of the epitope. This is the alpha chain, and it lays along a hydrophobic surface of the antibody. So the alpha chain's involved, but kind of not as intimately compared to the beta and gamma chains. And this explains why this antibody is able to bind strains that have extensions of the alpha chain, because this kind of has room to stick out this way. However, extensions of the beta chain, this galactose residue can be silated in Gonococci, and silation of this lactose residue would end up being here and really create a steric clash with the antibody. But in fact, this antibody binds to the vast majority of gonococcal strains, so it actually is viable as a therapeutic. So potential applications of this structure are: 1, to humanize the antibody as a therapeutic; 2nd, to engineer the mAb to increase its affinity for LOS, and also to understand the structural basis for peptide mimics of LOS. One mimic is shown here, and we solve the structure of this peptide bound to the same Fab as the gonococcal OS. So we can, it's not, you can't appreciate it from this slide, but this kind of defines the structural basis for mimicry by a peptide of the OS antigen. If you remember at the beginning of my talk, I showed you that MenB4C and MenZB from New Zealand contain the same OMV component. And after the retrospective study in New Zealand, studies in mice showed that the MenB4C vaccination elicited bactericidal antibody in mice, which I'm not showing here, and also accelerates clearance of vaginal colonization in mice. So the Kaplan-Meier curves in blue and red show the data for IP and sub-Q administration of the vaccines, followed by vaginal inoculation of the bacteria. And you can see significant acceleration of clearance in the vaccine groups compared to the negative controls. Following the retrospective study with MenZB, different groups have looked at the ability of MenB4C vaccine to protect against gonorrhea in humans. This table is from a meta-analysis of 6 different studies, which found vaccine effectiveness that ranged from 33 to 47% and risk reduction between 46 and 59% with relatively wide confidence intervals. And while all these are retrospective or observational studies, the range of values is pretty consistent with around 40% effectiveness. And as of 2023, MenB4C is recommended in the UK for persons at high risk of gonorrhea. So this vaccine is actually being used to prevent gonorrhea, and I think further prospective studies are needed to really know whether this affects. It looks reproducible from all these studies in the meta-analysis. I'll just show you quickly some data from the first prospective trial of MenB4C against gonorrhea. This used a 2x2 study design in which 2/3 of the vaccinated subjects also received doxycycline post-exposure prophylaxis for prevention of chlamydia and syphilis. The study did not show a statistically significant difference between the 4C MenB vaccine group that's shown in blue and the no vaccine group shown in red. The adjusted hazard ratio was 0.78 and the p-value was 0.06. And one limitation was that the primary outcome was PCR detection and not symptomatic disease. Therefore, it was possible that the vaccine might have had an effect on symptomatic disease, but not on asymptomatic carriage. And this, that's all I'll say about that. So in summary, I've shown you that meningococcal B vaccines elicit different antibody repertoires in mice and humans. They can be improved through informatic and biochemical approaches, and they might gain wider use through combination with conjugate vaccines in the men-penta approach. Finally, gonococcal vaccines might be achievable through a combination of outer membrane vesicles, such as the ones in men-B4C or men-ZB, overexpressed protein antigens, which could be put into an outer membrane vesicle, and such antigens have been identified through proteomics. And lastly, genetically stabilized LOS core structures, as in the structure I showed you a couple minutes ago. Thank you for your attention, and happy to answer any questions.
For your, you have a protein that has a high level of variability, these things about 80% homology or variability. So when you look to see where those residues are that are different, do they cluster in similar regions along the protein? Or are they distributed? You know, are they affecting protein function or the binding interface?
Yeah, so it's a great question. It turns out, and I think counter to what I might have expected, most of the sequence variation is in the factor H binding site. And the difference between the two subfamilies, which differ greatly in sequence, basically show that there are two different modes for factor H binding, one from subfamily A and one from subfamily B. There's also on the surface of the protein that faces the outer membrane, a pretty conserved part of the protein, and then also differences around the periphery. So I think it's like the differences are everywhere, but there's a couple of exceptions.
So when they're at that surface interface, are these variants like relatively neutral substitutions, like, you know, nonpolar for nonpolar or positive positive, or are they all over the place?
I think they're kind of all over the place. There's no, I mean, there's sometimes they're hydrophobic for hydrophobic, but really it's hard to generalize an answer, concise answer to that.
Much less question. It ain't quite. I was just really struck with so it sounds to me, if I follow this piece of your story correctly, that finding the protective effect on gonorrhea infections was kind of a random kind of discovery, right? Was it really random, or is that actually something that people routinely do? Because you can imagine that maybe there's other vaccines out there that are good for something else that we haven't even thought about. Is that a routine practice in vaccine testing, or was this just someone happening to look at the database and saying, oh, that's interesting?
Yeah, I think it was the second one. So I wish I was inside the person's head who thought of looking for this, because I mean, in hindsight, it's kind of logical because the bacteria are genetically similar. Sure. They're, you know, in the same genus. And so it's not surprising, but it's kind of surprising somebody thought of looking. And I think actually like years later, New Zealand apparently has a good database. It's a small country, and they have good records, good medical records. So they were able to do this. And also because they had an epidemic, almost everybody in a certain age group was vaccinated. So they kind of had like a perfect system to look.
And maybe as a follow-up question to that, this seems to me the kind of thing that came up then sort of randomly, that you could probably find an AI engine that will routinely do this for any database that you have out there with a vaccine level.
Yeah, interesting idea. Thank you all for coming.